So this year we always have a poster session, or a poster competition and session, and we're going to do a virtual poster session at the end of this morning segment and before we start our second keynote. But from all those posters, we also select the top poster for an oral presentation. And that oral presentation will be delivered this year by Dr. Dennis Watson. So congratulations, Dennis. It was a very competitive field. Dennis is a medical oncology fellow at University Hospitals. His poster was voted the best from an incredibly competitive pool, and here he is to present. He received his PhD from the National Cancer Institute, where his research focused on the translational development of interleukin-15 for HIV and cancer. Dennis's current research under the mentorship of Justin Lathia focuses on deciphering tumor and microenvironment interactions in glioblastoma and developing microbial extracellular vesicles for cancer immunotherapy. Dennis will speak about new ways to treat brain cancer with extracellular vesicles. Dennis.
Thanks. Thank you, Mukesh, for that very kind introduction. It's a pleasure to be here. And thank you for this opportunity to share with you some of our data on how we're using bacterial extracellular vesicles for glioblastoma immunotherapy. So glioblastoma, or GBM, is the most common primary brain cancer. Unfortunately, it's usually not very responsive to immunotherapy. And one of the reasons for this is that up to 50% of the tumor mass can actually be infiltrated by immune suppressive macrophages and microglia, as shown here on the right in this human brain tumor biopsy specimen, where you can see the massive infiltration of the tumor by these IBA1 CD204 double positive immune suppressive myeloid cells. So one of the questions that we had was, how can we actually target these immune suppressive myeloid cells in the context of glioblastoma? And given my background in extracellular vesicles during my PhD work, we thought about potentially using bacterial extracellular vesicles to target these cells in glioblastoma. So what are these extracellular vesicles, or EVs for short? So these are nanoparticles that are similar to liposomes, and they're produced by both prokaryotic and eukaryotic organisms. So in the context of bacteria, they contain a lot of immunologically active molecules like proteins, RNAs, DNAs, lipids that come from the bacteria. And depending on the actual strain that makes the extracellular vesicles, the EVs, you can get very different immune effects. So why even use these? So why use EVs to target myeloid cells? Well, the reason is that we had found, and others had reported this as well, that EVs have a natural tropism for myeloid cells. So on the left, you can see the association of mammalian EVs that are labeled and incubated with human peripheral blood, and most of those EVs would target monocytes in the blood, so the myeloid cells. On the right side, you can see what happens when we inject labeled EVs into a mouse, and this time they associate primarily with the liver and spleen, both of which are myeloid cell-rich organs. So the overall hypothesis is that we'll try to use bacterial EVs and their immune activating molecules as a way to interact with myeloid cells and other immune cells in glioblastoma, and hopefully augment anti-tumor immunity. So to do this, we needed a way of producing significant amounts of these EVs to do our both in vitro and in vivo work in the mouse models. And we adapted methods that I had worked on in the past, where we're able to culture several liters of bacterial culture, and then by sequential filtrations, as well as a pump-driven system and chromatography, we're able to end up with a few ml of highly concentrated material that is very, very enriched with these EVs. One of the first things we wanted to look at is what actually happens when we inject these EVs in a mouse and see where do they actually end up. And to do this, we generated a transgenic or a recombinant strain of E. coli that expresses nanoluciferase in association with the EVs. So we're now able to track these using a luciferase assay in the mouse. So in this experiment, we performed 3 injections of the EVs, and then 24 hours later, looked at different organs for where the vesicles would actually go. what we found was that the EVs in these experiments tended to go to the spleen, so again, something we had seen before. But interestingly, they also accumulated within tumors. And these are tumors that were implanted in the brains of the mice. And that would be the red column here versus the black column, which is the mice that did not get the EVs. You'll notice there's also a blue column there in the tumor groups. And those are mice that did not have a tumor, so they had a sham tumor, but did receive the EVs. But in those mice, we actually didn't see a lot of accumulation, suggesting that the presence of the tumor was actually important to enable these EVs to actually accumulate at the site. We've done some immune profiling as well, and I'll show just a snippet of that data here due to the time constraints. But essentially what we found was that when we injected these EVs into mice with glioblastoma, we saw a dramatic increase in the proportion of cells in the tumor that were leukocytes, that were immune cells. And you can see that in the bottom left there in the red group. And then just looking at the aggregate data here on the right, again, you can see about a 4-fold increase in the frequency of immune cells that are found in the tumor microenvironment with this treatment. Going back to the original hypothesis of myeloid cells, we looked at CD80 as an activation marker. And what we found was that both in microglia, which are the brain-resident myeloid cells, as well as tumor-associated macrophages, those are the ones that come from the blood. So in both cases, we saw an upregulation of CD80, suggesting that the EVs were activating myeloid cells in the brain tumor environment. And again, here is just the aggregate data showing about a 2-fold increase in the expression levels of CD80. The final piece of data I'll show is what we've seen on lymphocytes, a little bit of that. So looking at NK cells and CD8 positive T cells, so cytotoxic, potentially cytotoxic cells. We saw an upregulation of TIM3, which you probably know is an exhaustion marker, but is also an activation marker for these cells, suggesting that we were activating lymphocytes as well. In NK cells, we saw a similar activation of CD69. That's another activation marker. And we did look at PD-1. And interestingly, we did not see any changes in PD-1 expression with treatment with the EVs. So just to wrap up, what I've shown today in the brief time that we had was that bacterial EVs are able to accumulate in brain tumors. We've seen broad immune activation both in myeloid cells and in lymphocytes. In the future, we're thinking to combine with other lymphocyte targeting treatments like TIM3 checkpoint inhibition, and we're also going to test the effects of diverse bacterial EVs in our models. With that, I'd like to thank my mentor, Justin Lathy at the Cleveland Clinic, other collaborators, and then our funding support from the Harrington Discovery Institute, T32, and Velosano. So thanks. Happy to take questions.
All right. Thanks very much, Dennis. That was great. And I think I may have forgotten to mention, but I think it's really important that Dennis is a part of the physician scientist training program within our Department of Medicine. And that's overseen by Dr. Aaron Proweller. And I, prior to Aaron, had an opportunity to help start that program. And when you see people like Dennis, it gives you great confidence in the future of our institution in its scientific pursuit. So thank you. That was just beautiful.
Thanks.
A couple of questions for you, Dennis, that have come over through the chats. You are trying to activate immunosuppressive myeloid cells. And so a logical question is, do you see undesirable activation systemically, since these things travel all over the place? And we saw they went into the liver and spleen.
Yeah, that's a great question. So we see similar activation of myeloid cells in the spleen. So some of the same markers are upregulated. We've done a toxicity study that I can briefly show. So what we did in this experiment was we had different fractions of the purification, where the blue are the EVs that we isolate, and then different non-EV fractions. And then when we actually give these into mice, we see different levels of toxicity. With the EVs in particular in the blue, we see some decreased activity in the mice at about 2 hours, but they recover fully at 24 hours. Whereas with the other non-EVs, so other small proteins, et cetera, that the bacteria secrete, we actually see that the mice do not recover, suggesting that the EVs are actually less toxic than the rest of the bacterial secretome.
That's great. And I think if we can do maybe one more.
Yeah.
Is there any effect on survival in the, I guess, in the GBM models that you showed?
So I can show you the survival curve we have from our preliminary experiments here. So we don't see a statistically significant increase in survival with monotherapy. I think there are a couple reasons for this. So one is there's a model limitation where we actually see the tumor mass growing because of the immune cells coming in. So because you're in such a constrained space, that turns out to look as like a loss of survival. And on the other hand, we did see the upregulation of checkpoints on the T cells, which is exactly why we think that moving forward, if you do a dual approach, you may get a more synergistic effect. Yeah.
Got it. All right. It's also a good sign of a young scientist that has backup slides that answer all their questions that the audience has. Well done, sir. Thank you.
Thank you.